talk about operons and the way they work in controlling transcription and translation of proteins in prokaryotes. So first what we're going to talk about are things called transcription factors, and these are simply proteins that determine whether or not a gene gets expressed. And there's two types of these that we can have. We can have either a repressor. A repressor is going to repress transcription, or in other words, it's going to stop transcription from occurring. We can call this negative regulation when transcription is blocked from happening. We can also have an activator. An activator is something that's going to turn transcription on, and that would also be called positive regulation. So when we look at the two of these, they're basically just opposites. A repressor is going to turn off transcription, which is going to turn the proteins or the genes off that we're looking at, and the activator is going to turn the genes on that we're looking at. In order to understand how transcription works for humans, first we're going to look at prokaryotes. Prokaryotes, these are single-celled organisms, no nucleus, no membrane-bound organelles. That should sort of be where your brain goes when you hear that term pretty quickly. And when we talk about something that's that simple, without a nucleus, without membrane-bound organelles, conservation of energy is going to be extremely important, and efficiency is going to be absolutely key for the survival of the bacterium. Prokaryotes basically work where if they have something, fantastic. They'll use it, they'll divide, they'll do whatever they need to do to go through all their life processes. If they don't have something, then they won't use it. And ideally, they have genes that can make what they need, and they just try to use what they need. Once they have enough stuff, they will simply divide and go ahead and do whatever they need to do to go through their life processes. A specific bacterium we can talk about is E. coli. You may hear of E. coli from food poisoning cases and tainted lettuce, all of those kinds of things, but realistically, you have a ton of E. coli living inside of you already, and they live down deep in your intestines, and they just sort of survive in that environment. Now, the environment within your intestines, every time you eat something, you change that environment. So the E. coli are very sensitive to what's going on around them. And one thing they're really sensitive to is something called lactose, which if you look at the end of the word, the OSE, you should be able to tell that that is a carbohydrate. Well, catabolism means the breakdown of lactose. So the E. coli will break down the lactose and use that as a energy source, because lactose is what we call a beta-galactoside. Beta-galactoside is a combination of a galactose monosaccharide and a glucose monosaccharide, and those two things form to create the disaccharide lactose. E. coli requires 3 different proteins to break down the lactose and use it efficiently. The 3 proteins are listed here on the page. Beta-galactoside permease, that's going to allow the lactose to enter the cell. There's receptors on the outside of the bacterium that sense that it's coming, and the beta-galactoside permease will bring that lactose into the cell. Beta-galactosidase is going to break down the lactose into the individual galactose and glucose monomers, and beta-galactoside transacetylase is going to transfer some acetyl-CoA groups, and the scientists don't really know why, and you know what? That's okay. So if the bacterium is in an environment that lacks lactose, well, those proteins aren't really needed, and since it requires energy to make proteins, the prokaryote simply doesn't make those proteins. It has those genes to turn those proteins turned off, and if those genes are turned off, they aren't being used. As soon as the E. coli is added to, as soon as the lactose is added to the environment around the E. coli, well, then the E. coli is like, okay, I guess I can use this stuff now. So now we need to turn that stuff on and increase the amount of proteins that are being made to help break this stuff down for us. Now, as we move forward, there's a couple terms we need to understand when we talk about lactose catabolism. First is an inducer. An inducer is kind of synonymous with the activators. This is a compound that's going to turn a gene on. It's going to stimulate transcription, which is going to create the proteins that are needed to digest the lactose. Inducible genes are genes that are normally turned off, but in the presence of an inducer, they get activated, they get turned on. Now, a little different is something called a constitutive gene. A constitutive gene is one that's just sort of in the genome of the bacterium and is always being created. It's always on. I always liken it to 7-Eleven. It's a 24/7 thing that's just sort of always going. So in our case, the constitutive gene is going to be the one that makes the repressor that's going to keep the lac operon off before we can turn it on, and we'll sort of get to that in a couple slides here. One important thing to know about bacteria is that in order to maintain efficiency, they'll use something called an operon. An operon is a group of genes that are encoded with one promoter. If you remember from our talks about DNA transcription and translation, a promoter is a sequence of bases on the DNA where the RNA polymerase knows to grab and start the transcription process. So for bacteria, they'll have one promoter and multiple genes after that promoter. So the RNA polymerase only has to attach once. Again, this is going to be increase the efficiency for the E. coli or for the prokaryote so that it can conserve energy and do what it needs to do. An operator is also a segment of DNA, similar to the promoter. It's actually right next to the promoter. And the operator's job is that's the place on the DNA where the repressor will actually bind to. Now, hopefully you remember that a repressor is a protein that is going to regulate transcription, and repressors repress transcription, meaning they turn it off. Now, if you have a gene that is normally turned off and can be turned on, then we would call that an inducible gene or an inducible operon. The operon that we're going to be talking about is a lac operon, which is short for lactose, and that is an inducible operon because, like we said before, the proteins that are needed by the E. coli to digest the lactose aren't needed unless it's in an environment where there's lactose surrounding the bacterium. So as long as there's no lactose present, then the bacterium doesn't need those proteins, so those genes are turned off. As soon as the lactose becomes present, though, then those genes get turned on or induced. So that's why we call lac operon inducible. When we are looking at the lac operon, well, there's also a repressor that's being transcribed that's going to work on the lac operon. The repressor comes from what we call a constitutive gene. Again, that's the one that's 711. It's always on, it's always working. The repressor that's created from that constitutive gene is going to bind to that operator site. Remember, if the repressor is bound to the operator, that means that the RNA polymerase can't grab onto the promoter and move through and transcribe. The repressor is going to block that, kind of just similar to a roadblock. So in order to turn this thing on, we need to have an inducer present. The inducer is going to help remove that repressor. In the case of the lac operon, allolactose is going to be the inducer. The allolactose will bind to the repressor, and through allosteric regulation or allosteric inhibition, that repressor is going to change shape, because that's what allosteric regulation means. A molecule binds to a protein and it causes a protein to change shape. Once that protein changes shape, it moves off the operator and allows transcription to take place. If we look at this picture, we can see that you have the PI is a promoter for the constitutive gene, which is the lac regulator, the lac repressor gene. So that I, the yellow segment on the DNA here, is going to be the segment of DNA that codes for the repressor. The repressor attaches to the O segment of the DNA, which is the operator. The PLAC is a promoter for the lac operon, and ZYA are the spots where we have the three genes, the beta-galactosidase, beta-galactoside permease, and the beta-galactoside transacetylase. So that orange thing on the top that's your RNA polymerase, isn't allowed to attach to the promoter and move past and transcribe the ZYA genes. So it's sort of getting stuck. Once lactose becomes present, as you can see at the top of this picture, lactose attaches to the repressor. It causes the repressor to change shape. As it changes shape, that opens up the genes and allows RNA polymerase to attach to the promoter of the lac operon and zip its way down to the right through the Z protein, through the Z gene, the Y gene, and the A gene. That creates an mRNA transcript, and that transcript of mRNA will then get coded into proteins that will allow lactose to be broken down and used as energy for the E. coli. So the lac operon is just a quick review. The repressor gene for the lac operon is constitutive. there are 3 proteins that are used in the digestion of lactose, and unless there's lactose present, those 3 genes are turned off. Our inducer for this entire thing is the allolactose. The allolactose is the thing that's going to allosterically regulate the repressor that is normally blocking transcription to happen. Now, if we have operons that can be turned on, well, you better believe we have operons that can be turned off. And the terp operon is one that is what we'd call repressible, or one that can be turned off. TRP stands for tryptophan. Tryptophan is an amino acid that the bacterium can use. Well, the bacterium actually has multiple sites on its DNA that code for proteins that can build tryptophan themselves. Well, if there's no tryptophan present, then the bacteria will make tryptophan on its own, and that amino acid can be used to make other proteins. If there is tryptophan present in the environment around the bacteria, which, again, we talked about, the environment around the bacteria can be changed based on your diet, well, then this operon is going to be turned off because the whole purpose of the prokaryote is to remain efficient. If it has tryptophan, it doesn't need to make its own, so it's just going to take the stuff from the outside. And tryptophan is going to act as what we call a co-repressor. A co-repressor is going to be a molecule that binds to a repressor that's turned off, and it's going to turn the repressor on. Once a repressor opens up and is allosterically regulated, it's going to attach to the operator and turn off those genes that the bacterium doesn't need to use. So when we look at this picture of what's going on in the terp operon, on top you see the DNA with the blue and the yellow bars. Well, that creates mRNA. That mRNA creates an inactive repressor. The repressor isn't doing anything. Since a repressor isn't attached to the DNA on the bottom with the long darker blue, the P-terp is a promoter for the terp operon, the O is the operator, and then EDCBA are the different genes that create the enzymes that allow tryptophan synthesis to occur. Well, since there's no repressor on the operator, RNA polymerase can attach to the terp promoter and go down and transcribe the mRNA transcript, which will eventually be translated into the enzymes used for tryptophan synthesis. If you look on the right, when tryptophan is present, tryptophan is going to act as a co-repressor. If you look at the word co, means with or together with. If it's together with the repressor, once it gets on there, it's going to activate that repressor. That repressor then opens up and is allowed to attach to the DNA at the operator site of the terp operon. Once it attaches to the operator, the RNA polymerase can no longer attach to the promoter and transcribe down through the EDCBA genes. Instead, it gets blocked, and that operon has been turned off. It's been repressed, which is why we call the terp operon a repressible operon. So, and this is words of what we just saw. The repressor complex will bind with both items, and they will attach to the operator, and that's going to block transcription. Since it's blocking transcription, we have now turned those genes off, or we've repressed them. This is only going to happen if there's enough tryptophan for the bacterium to use, though. Again, this is just something that bacteria are trying to remain efficient, so if they don't have this, tryptophan is something that they need. It's an essential amino acid for them, so they'll just make it on their own. But as soon as they get a heavy enough supply, they're all good. When we look at the two systems, we can compare them. Inducible systems, well, you have an inducer that's going to interact with the repressor. In the repressible system, you have a co-repressor that's going to interact with the repressor. The inducer is going to turn the repressor off and move the repressor away from the DNA so the transcription can happen. A co-repressor is going to turn the repressor on and allow it to attach to the operon, which is going to block transcription. In both of them, the repressor is changing shape. One of them, the repressor leaves the DNA, that's the inducible system. One of them, the repressor binds to the DNA, that's the repressible system. Both of these systems allow for gene expression. One of them turns gene expression off, one of them turns gene expression on, and the reason we do this is because efficiency is essential for the prokaryote. Protein synthesis does require ATP, so the more ATP the prokaryote can save, the better for them. Hope you enjoyed this, and I hope this makes a little bit of sense. We'll be talking again soon. Have a good evening.